As Ala said, we're going to be talking now, similar to what you just heard from Anna in study design, we're going to talk about sample collection. So the objectives for this talk, and by the end, hopefully you can identify at least some of the requirements, the general requirements for specific sample types. And most importantly, if you take nothing else from this talk, understand the importance of consistent sample collection for using in metabolomic studies. So as you've heard from Chuck and Anna, the metabolome is influenced by many different factors. These can be genetics, they can be age, they can be different drugs that you're taking, it can be nutrition, your food that you're eating, it can be influenced by even things like lifestyle choices, exercise. Because of this, you have to be extra careful with metabolomic studies in collecting your samples and designing your study so that you limit the effect that these have on your final data output. So this talk is going to be given from the standpoint that you have not done your sample collection. We're going to talk primarily about the optimal methods. I recognize that that generally is not the case. You don't come here because, you know, in the future you might sometime think about doing this. So many of you I know have either already collected or have started your collection. So at the very end, we'll talk about a few strategies that can be done or can be used if maybe you didn't follow, you know, everything exactly, things like that. All right, so where are we in the workflow? We're at sample collection, obviously. It goes hand in hand with consultation and study design. Some of the common types of samples, as you heard from Anna, are cell culture, different biofluids, and tissues. We're going to focus primarily on some of the most common, so cell culture, different biofluids such as blood, plasma, serum, and urine, as well as touch on tissues a bit in the generic sense, not down to specific individual tissue types. So for these samples, I'm not going to go over every single aspect about how to collect them, how to process them, things like that for giving to us. We have a document, a PDF document that details this in more specifics. You can access this either through our website. If you click on the project request link, there's another link to sample collection guidelines, or you can cut and paste this into your URL, the bottom thing here. It's also been put into your folders, so there's many different ways to access it. Finally, you can email us, and we'll be more than happy to send it to you. It's a much more detailed protocol, but because you can just read it, there's no point in me going over all of it. There are, however, some common themes that you'll hear throughout this talk. The first, and this is something you'll hear throughout the entire workshop, metabolomics, because of all of the factors that can influence and because of the diversity within the metabolites themselves, a lot of this is dependent on your experiment. So there's not one uniform anything in metabolomics. So it's very dependent on your sample type as well as what your objectives are. Consistency is key. Please, please, please try to understand from this talk how important it is to be consistent. Keep it cold. No matter what sample type you're using, we recommend that you keep it as cold as possible. This includes processing on ice, freezing it as soon as possible, shipping it on ice, on dry ice. And then consult. Do not be afraid to ask questions. Do not be afraid to email us. Hopefully, we can help you prevent some of these outside influences that you then have to deal with afterwards. So if you get rid of it at the beginning, it makes your data analysis much easier. Okay, so for cell culture is frequently used. As Anna said, you need to determine the best conditions for your cell type. So whether you want to collect your cells when they're confluent, whether you want to collect your cells as they're growing, what growth phase. One thing to remember, though, is that each sample should be at the relatively same confluency, so the same number of cells, because something as simple as that can influence your final data. It can influence metabolite expression. Where this really comes into play is if you, say, treat your cells with a compound. That compound frequently will affect proliferation. It can affect cell death. These are things you need to think about and maybe do an experiment up front to see how much this is going to impact your final output. Because what you want to see is how, say, that compound is going to influence your metabolites, not how a different growth phase is going to influence your metabolites. We recommend that you do freeze your cells in liquid nitrogen, whether you have suspension cells or adherent cells. If you have adherent cells, though, we typically recommend that you quickly rinse your plate and then pour your liquid nitrogen directly on the plate itself. There's no need to scrape them. There's no need to trypsinize them. Just go ahead, pour it on. When it evaporates off mostly, cover your plate up, put it on ice until you're ready to ship it or deliver it, or whatever the case may be. Some cautions to think about with cell culture. Try to remember to stagger your start time for different plates. Frequently with cell culture, we use relatively short time periods. So if you're going to treat with a compound, it might be for 15 minutes. If you forget to stagger your start times, your last plate that you collect could be off by a bit. So remember to do that. Think about your media type. So this really comes into play if you're going to do a fluxomics experiment. Frequently, medias have additives to them, and it's important to know what is in your media. So in this case, in this picture, it's a picture of RPMI media, and it says on there, which of course you cannot see, plus glutamine. So if you were interested in doing a fluxomics experiment with a labeled glutamine, this would cause a problem. So this is a place where if you are going to do an experiment of this type, which you'll hear about more tomorrow, really consult with us, provide us a list of what is in your baseline media so that we can make sure there's no interfering factors that are going to influence your results or limit the usability of it. And finally, even if you don't plan on analyzing it, we recommend saving your media for analysis. That way, if interested, if need arises, you can look at the exometabolome or what is secreted. You may not think that's important, but then you find something different to what you think, and it may just be that it's being secreted, not that it's not there. And so it may become important, so it doesn't hurt to store it for a little bit. So go ahead and save that. Okay, switching to biofluids. As I said, if nothing else, be consistent. So the tube that you collect your plasma or serum in can greatly influence your results. So here, what we're looking at on the right is a figure, and just up here, this is the influence of the tube itself. So this is no sample, this is no plasma, this is no serum, just differences caused by the type of collection tube. And then when you put your sample in this case it's plasma, you need to be able to detect the signal from your sample over the background signal. So for this reason, we prefer to use EDTA collection tubes. However, if you've already started collecting your samples in a different type of tube, such as lithium heparinate, continue doing that. Try not to change your sample, as that can cause differences in your final data. Also, after you collect your sample, we recommend that you aliquot it out immediately before freezing it, preferably into chilled tubes, at least into tubes. The reason for this is that freeze thawing can have an effect on the expression of many different metabolites. So as you can see in the bottom here, this is L-carnitine. And what they did in this study was to take a fresh sample and analyze it, or aliquot it into different tubes. They then froze it and thawed it once, so that's the one time, or froze it and thawed it multiple times, and then looked at expression of multiple metabolites. And as you can see clearly here, there is a change in metabolite expression. And we see this in different classes as well. So the best way to do this is just to pre-aliquot it instead of having to thaw your sample, take out what you need, each time you want to do some type of analysis. We also recommend that you, as soon as possible, do freeze this, preferably in liquid nitrogen, and then store it at -80 and transport on dry ice. So this study shown here was performed to simulate the effects of different shipping conditions on plasma metabolite expression. And I'm only showing you one metabolite here, arginine. However, if you go to the paper, they actually did this for many different metabolites across a variety of classes, sugars, amino acids, lipids, and they found similar trends across many different classes. And what that is they took a sample and immediately froze it. So that's your 0 hour. They took an aliquot of that sample, put it on the lab bench for 24 hours at room temperature, or they took aliquots and put it on cold packs to simulate refrigeration, so 4 degrees, for 3, 6, 6 or 24 hours. And, pretty much regardless of the class type, if you kept it at room temperature for 24 hours, there was a marked change in metabolite expression across a variety of different metabolite classes. But even something as small as a 3-hour refrigeration or, shipment on cold pack or on just wet ice instead of dry ice can change the expression of some of these metabolites. Another thing to think about with blood is hemolysis. So this study was done to look at the effects of hemolysis on blood on metabolite samples, or on a metabolite expression. So what was done was blood was collected. It was then aspirated through a needle to either induce moderate hemolysis, or strong hemolysis. And they looked at fold change of untargeted data. And they found that regardless of whether it was a moderate level or a strong level, that there was a change in multiple different peaks, so multiple different metabolites. So it's best just to try to avoid this altogether. Some other cautions: really remember to limit your processing time, especially if you're taking multiple samples. Try to keep individual samples on ice if you're not actively working on them, and preferably put them in liquid nitrogen as soon as possible. And please, please, please be consistent. Okay, so with urine. Urine is another biofluid that we frequently see. We do recommend a 24-hour collection if possible. However, with patients, this is not always easy to do. So in that case, we recommend a first morning collection. With patient samples, we do recommend that you collect midstream in a clean catch fashion. And then if they're collecting at home, they can store it in the refrigerator short term until shipping it to you or bringing it into the lab. And with animals, it is preferred to use a 24-hour collection in metabolic cages and to collect the sample on ice. So the study depicted here is from rat urine, where they took rats, they put them in metabolic cages, and they collected for either a 24-hour period or a 4-hour period. And they collected the samples either on ice, so they had a bucket of ice under there with the tubes in it, and those are the closed black circles and squares, or they collected it at room temperature, which are the open or white circles or squares in the figure. And you can see that there is a difference. These groups separated out between the 4-hour collection and the 24-hour, and they segregated more nicely with the 24-hour, so they were tighter together. And then furthermore, even within that, you can see there were changes between those collected on ice and those collected at room temperature. So we do recommend that, if possible, to do a 24-hour collection and to do this on ice. For processing your urine, you heard from Anna that we do recommend you add sodium azide to the tube. So how we do this is that we normally estimate the amount of urine that we frequently collect, and we add sodium azide to our tube before collection to approximate concentration of 2.5 millimolar. And this is done, once again, to prevent bacterial growth. Next, you do want to centrifuge the sample. You want to precipitate out the sediment. And when you draw the urine off to collect it, make sure you leave a small layer in between your pellet and what you take. You can get a little bit of mixing there sometimes, and that can influence. So it's best just to leave a little bit there. And you normally have more than enough urine for any type of analysis that you're going to need. So it's fine to leave a little bit in there. As with plasma or serum or whatever, please aliquot it into tubes, because once again, freeze thaws can have an effect on your metabolite expression, and store it at -80 as best you can. Obviously, if patients are collecting this at home, they can keep it in the refrigerator for short term, but as soon as possible, go ahead and store it at -80. Some cautions to remember. Sampling, we frequently think of sampling plasma after meals, medications, that, we see how that could have an effect. That will also have an effect in your urine metabolite expression. So Anna showed you a figure of dietary effects on metabolite expression in the urine. This is after medication. So here on the bottom, you're looking at an NMR spectra. This is of a patient that was taking no medication. They then took either acetaminophen or ibuprofen. In collecting urine after that, you can see if you look just even at the expression of citrate, which is right here, that taking acetaminophen had a drastic change in the expression of citrate. So something as simple as, you have a headache, you may not think about that, but just remember that this can be reflected in your urinary samples as well. Okay, tissues. Tissues, it's hard to go into too many specifics on tissues because there are so many differences amongst different tissue types. So the amount that you're going to need is going to depend on what type of tissue you're collecting, as well as what you want to do with it, what type of analyses you want to run. Regardless of that, though, it is important to use a consistent sampling time and method. And because there is so much variability between tissues, try to really consult with us, because there may be something inherently unique to a tissue, like skin versus kidney or versus liver, that may present unique challenges, and that would be important to know up front. However, regardless of what type of tissue you have, please snap freeze it as soon as possible and store it at -80 and ship on dry ice. Okay, so even with tissues, it is incredibly important to be consistent. As we said many times, and I'm sure you'll hear again, the metabolome is affected by a variety of things. These can be diurnal variation, whether they've eaten recently, or even things like anesthesia type when you're talking about animal models. And you'll hear from Charles later about this, a study that was done to look at different anesthesias on tissue metabolite expression. What I'm showing you here, though, is actually a study that was done to look at the expression of different lipid classes under control of their circadian clock. So they took animals and they changed the time at which lights came on. So regardless of what time the light was turned on, they called that 0 hour. 12 hours later, lights went off. And they found that many different lipid species and individual lipids within classes changed, and they had a peak expression time dependent upon when the lights were turned on, so under control of their circadian clock. So with the orange dots here, these are different tricylglycerols, and their peak expression occurred 8 hours after the lights came on. So typically, if you're going to do an animal study, your animal room tightly controls what time the lights come on in the room, yes. But if you say start your experiment and you analyze all of your control animals, or you collect tissues from all of your control animals, you then break for lunch because everybody gets hungry. You then come back and you collect from your disease or treated animals. You could inherently be inducing bias in your results solely because different metabolites or lipids can be under control of something like the circadian clock. So in this regard, we recommend that you actually alternate your groups, whether it's one control, one disease, or two control, two disease. But you can help limit some of that external bias that you could put into your samples, because so many different things can influence your final measurement. With animal studies, we also recommend that you perfuse the tissues. So prior to taking them, we recommend just using something like PBS. Blood within the tissue can alter your metabolome. You may get more of a signal from the blood than the tissue itself. So if you wash it out, then you can limit that effect. Finally, if your tissues need to be dissected, please do that before freezing them and sending them to us. I would not like to make sure that we're responsible for knowing which exact cell type or part of the heart or whatever you would like to analyze. Some general tips in terms of your experiments. Please write on your tubes themselves with permanent marker. Do not just put labels on them. So everybody, I think, has used cryotags. They tend to fall off in liquid nitrogen. Nothing is more frustrating than performing an experiment, getting all of your samples, pulling them out of the liquid nitrogen, and realizing 3 of them no longer have labels on them, and you just have to throw them away. It's really, really, really frustrating. So even if you just write, 1 through 40 on your tube and you have a master key, that is fine. But please write something on the tubes themselves so that, worst case, you can still use them. Document, document, document. Because so many things affect the metabolome, it's really hard to control for all of them, particularly when you're talking about clinical samples, right? So the more information you have can really help you when it comes time for your analysis. If you see something that's not behaving the way you thought, a metabolite or whatever, you may be able to pinpoint what is causing that effect if you have information. So please, note down sampling sites. So if you have, a multi-center study, or even if you have multiple people collecting your samples, have them initial it, or on in your notebooks, make sure that's noted. The exact collection method that was used, or the exact time that it was taken, note that down. If you have multiple people or multiple sites collecting samples, please develop a very, very detailed sampling protocol. Nothing, in my opinion, should be overlooked. When you think back to what we said about the urine, make it clearly stated that you leave a small layer over the pellet, and you take off, the top. Leave areas, leave a line if, you say you collect from this vein, but if for some reason you can't for a certain patient, there's a spot for them to write down where it was taken from. You can really help to limit variability induced by non-consistent sample collection if you have a very detailed protocol that is easy to follow, because not everyone may understand how important some of these factors are. Once again, please consider your groups during collection. So do not take all from, one group followed by all from another group, your control, diseased, what have you. And then finally, regardless of what you're doing, consistency is incredibly important in these types of experiments. And please, please, please consult with us if you have any questions at all. So to go back to consistently be consistent. Small changes can have very large effects, and it's just best to get rid of these up front by being consistent. Things like changes in diet. We've had cases where, our animal room has a standard chow diet for the mice. It gets back ordered, so they buy it from a different vendor. The source of fat in that diet might change. It may not seem important, a certain type of oil or, 5% difference. But if you ran a pilot experiment with 3 animals and it looked great, and you want to go back and do, 7 more animals or 15 more animals or what have you, it will be very hard to compare those results if your diet has changed. Changes in sampling site. So this figure here depicts that. Plasma samples were taken from rats, and they were taken from either the abdominal aorta or the jugular vein. That was the only difference. So they were collected from the same animals from 2 different sites. And you can see that they were able to separate these samples solely based on where blood was collected from. This may not always be easy to control, but particularly in your sampling protocol, you can have a recommendation and try to keep that consistent. And then finally, changes in sampling time can have an effect, as we talked about with the lipid study under circadian control. Okay, now normally everyone's scared, and you've already done it, you're frustrated, it's like I can't use my samples because I didn't follow all of these things. So what to do if maybe you were not able to have, optimal sample collection? The first thing is just to consult with us. Be as honest as you can, tell us what was done. Hopefully you have some information, and this is where information really is important. Many, many studies have been done, and this is where I say look to literature. I showed you examples from from several different studies that were undertaken solely to look at the effects of different sample collection methods on metabolite expression. They're not all negative. That's what we highlight. It's sometimes class dependent, so it may be that, what you want to look at, it's not such a big deal. And even if you didn't follow maybe the optimal conditions, if you consistently didn't follow them, then at least that effect was across your samples, okay? It may be that you want to run a pilot experiment. Typically, you want to run a pilot experiment, but you may need to do one just to determine the effects of your sample collection. So if you collected urine from patients and you asked them to bring it into the lab within 48 hours, yet 30% of your patients said, oh, we shoved it to the back of a fridge, we totally forgot about it. 1 week later, they bring it in. 2 weeks later, they bring it in. It's pretty easy to develop an experiment to take urine, aliquot it out, keep some, in the refrigerator for 2 days, some in the refrigerator for a week, some for 2 weeks, perform a trial experiment to see what effect that has on your metabolites of interest. Once again, though, this is where information is really important. And then finally, you can compare your data analysis with your sample information, assuming, again, that you have information available to you. It may be that, there was something small in your sample collection that was overlooked, but that, caused an effect, and you can then either analyze those groups separately, or you can throw out that one feature that's throwing it off. So there are ways if maybe your collection was not optimal. However, the best way is just to try to limit this up front by being consistent in your sample collection methods. So these are the references I primarily used to put this talk together, and I'd be happy to take any questions. Yeah. Mm-hmm. Yes, so you're always more than welcome to do that. If you're looking at steady state metabolites, the culture media is more dependent on what you want to get from your cells. So, how much serum you need to grow that, might not have an effect on the metabolites. Particularly, though, when you're looking at doing a fluxomic study, that is when the actual ingredients, and yes, going to the website of the manufacturer, Gibco or Invitrogen or I think they're the same, but whatever it may be, going and you can print out the PDF, or you can save it and email it as an attachment. That is a great idea. It won't take long, to look at that for us and say, yes, you're fine, or it'd be better if you could find this media without this, or if not, create your own basal media, by adding these things together and leaving out this one component. But yes. The media? I'm sorry, yes, do freeze your media. So the question was, with cell culture media, do you need to freeze it in any way? Yes, freeze it, keep it at -80. In case, if you already know you want to analyze it, go ahead and ship it with your cell pellets or your plate of cells if you have adherent cells. If not, store it for a little bit until you get your data back and you can see if that's something that would be interesting to you. Yeah. Yeah. Yes. So the question was, how much tissue do you need? That is something that is frequently dependent on what you want to analyze. So, the question was, if you have a human biopsy, you typically do get a small amount of tissue. So a lot of that, unfortunately, comes down to what type of analyses you want to run and how many types of analyses. So many of our assays can be complex together. So if you're talking about targeted assays, there are ones that we can use the same sample to run multiple assays with. And then, we just need a small amount. I've used human biopsy samples from the kidney needle biopsy, and it's been enough. But once again, it just goes back to how many things you want to get from that sample, and knowing up front to what you want to get from that is really important in interacting with us, letting us know, so we know how much of that we need to save for each assay. And if we're not sure that it's going to be enough, the best thing would be to get one sample, maybe not that's important to your study, but just one sample that we could try and see if we are unsure. But typically, we've done most of these assays enough that we do know how much we need. But it is very, very dependent. Yeah. Right. So the question was, with patients, how long ahead of time do you need to control for many of these factors, such as diet, activity, maybe things like that? Is overnight enough if you're taking morning samples? I would say that some of this comes down to, once again, what you want. We do experiments, and I'm not a clinician, so I will defer to Chuck here, where we feed a standardized diet the evening before and then collect samples that have been fasted since that. We normally feed at, I think, 4 p.m. But as to specific hours,
yeah. So I think Kelly says it's right. It depends on what you really are going after. And I'll talk about this a little bit in the next talk. But it's you have to think about what's the question you're asking and how much difference will it make if a diet is different. So with George is not here, but we can see changes in metabolome after one meal, once they come back to a fasting state. So doing dynamics of metabolomes and untargeted, about 4 or 5 hours, people will come back down to where they were. But if you give them a polyunsaturated fatty acid diet, for instance, and I'll show this a little bit more tomorrow, is that even after 2 days, there's a massive reordering of the metabolome, but only part of the metabolome. And so again, it comes back to the questions that you're trying to answer, but consistency in whatever you do is so much more important than anything else. And I think you're going to hear this time and again, that the biggest variability still is an inherent biological difference between people, but the second biggest variability is the variability in the collection process. So no, it's not a satisfying answer, but it's like everything else that we do in biology, that there's a lot of different variables. The problem with metabolomics is metabolism is fast.
Sure.
Wait till the next talk.
No, go ahead. There's a little button there.
All right. Chuck, maybe you're going to address this later, but have you guys noticed any differences? I guess this is important for human subjects with like local lidocaine administration on your biopsy sample metabolomic outcomes.
Go ahead.
We see lidocaine in the metabolomics trace for sure, and every once in a while we get samples from some usually cranks, your samples, but we get odd peaks and we just don't know what they are, and is it because there's some contamination in the things that we inject in people and things like that. Again, nothing's perfect, but as you know, it actually tends to work.
Yeah, and once again, be consistent. If you're going to give it, give it.
Anybody want to do no anesthetic muscle biopsies?
Yeah. Yes.
So I was wondering if you could comment a little bit more about hemolysis. So I noticed that you said if you're collecting tissues, it's best to perfuse with PBS.
Mm-hmm.
So I'm just sort of trying to visualize that, because some of the tissues I'm interested in are going to be quite bloody, liver and placenta have these sinusoids. And
so this is with animal studies.
mm-hmm.
Are you talking about animal studies or
yeah, I know
patient
that
biopsies?
was on the slide for blood samples, but I was just curious if it also.
I'm sorry. Are you
Or
you
maybe it was
taking
on the slide for animal studies. Yeah.
So the perfusing with PBS is
Yeah.
is in an animal study.
Yeah.
That
How
would be
important is that?
a little more different to do.
I guess that's why I'm wondering, like, what's the
Right.
danger of hemolysis, and how important can it be in founding a study?
Right. Um, so this, so every, once again, everything you're going to hear sometimes it's dependent on what tissue you have. It's dependent on what your analysis is. So in terms of perfusion, that is somewhat dependent on what tissue you're taking. So liver, is different than skin. I don't know how you're going to
So
perfuse skin.
I'll let Sue speak as well. So Charles here, yeah, Charles is here, and he's done some stuff, and maybe he can comment as well. But when we worry about blood contamination of tissues as well. If you do a nice blotting, and it seems like it doesn't make a huge bit of difference, at least for liver tissue. Charles, am I speaking out of turn? But so the other thing I think you should remember and realize is that if you have a cell that's going to be taking up and or putting out metabolites, the blood metabolite concentration is likely going to be very much related to what the intracellular concentration is going to be, because this is a local exchange that you're seeing, especially in liver, and so some of the TCA cycle intermediates that we see rapidly enter and exit the cell, and so you're going to get a relative level. It may not be exactly what the internal concentrations are, but these things are moving very quickly.
So just one other point. I think the idea is basically for you to maybe to do a pilot experiment and to figure out where your tissue actually behaves. So for example, in the kidney, we have done the pretty similar to what Charles was saying, and the variance of the data is substantially lower when you actually perfuse as opposed to not perfuse. So I think it would be an important thing for you to look at, because that's the tissue that we analyze in my own lab most often. And I do think that it's an important thing for you to actually think about what tissue you're measuring, and just do one pilot experiment, do it either way, and see a few samples and see what your variance of your data is. And the timing is important too, as Charles was saying. You do want to make sure that you don't want the completeness of your perfusion overtake. That you basically, if you spend like 45 minutes trying to perfuse an animal, it kind of defeats the purpose of ex vivo metabolism going on. So you want to do it in a rapid manner.